workshop, we'll deal with doing some GWAS, looking at how to do linkage analysis, kinship, PCA, and so on. So now from the genotypic portion, we're going to add in the phenotypic data and see how we can do some analysis here. And this, I hope, will be useful for mainly the grad students who are working on genetic mapping on a diverse panel, plus also postdocs if any of you are here. First thing we will do is import some data. So I'm going to do open as, make best guess is okay. And as I said earlier, if you click on these sort positions, it'll automatically sort the genotypic positions for you in case they are not already sorted. So I have 3 files here, as you can see. This one is the genotypic file, the intermediate wheatgrass C3 cycle 3. And the 5% there, I just sort of name it to tell myself how much missing data was there to begin with. And after that, it was imputed using the KNN, the LDKNNI method that you just learned. And it's in HapMap format as indicated by the HMP. And then we have the phenotypic data. And there's the Q matrix I obtained from the structure. Of course, we're not going to go through that today. But if anyone is interested, just let me know. You can run it on Windows, probably Mac too, but I mainly do on MSI. It takes quite a bit of time, especially if you want to do 50K or 100K iterations. It's not a difficult program to figure out, but there's a lot of parameter options and so forth. However, I do have the output in case you want to see what the output looks like. So let's go ahead and select all 3, and all of these are also on the GitHub page. I will try to go as slow as possible, assuming that you are following along. If not, these files will be there for you to practice on at a later point. So you can also import this individually, by the way. I'm just going to do all at a time. Give it a second. Oh, maybe should have done one at a time, huh? There we go. So we have the genotypic file here. It's not colored, and I'll tell you why, as you saw in the previous presentation. Here's the Q matrix. Let's go through this file first, because I will not be coming back to it later on, because we're just going to do PCA and kinship for the GWAS today. So the Q1 and Q2 are the so-called Q values assigned to each individual, and that's based on their relationship to other lines or other genotypes in this panel. I picked 2 based on what the program suggested. Some of you might have 7, some of you might have none. It all depends on the population and how much of stratification there is. But for this population, we have to define that the K number, or the subpopulation number of 2 was the most optimal for this data set. Again, it's just a numeric matrix assigned to each of this line in the whole population. It's all it is. We're going to instead use PCA today. But if you want to use it, I will show you how to include it in your analysis. But for now, we're just going to keep it as it is. So the reason why this is not colored as before is it does not treat it as a HapMap file, because we imported all at once. It happens for some reason. So I'm going to delete that. Yes. Then go back, open as. And then here, instead of now making the best guess, I'm going to open as HapMap and just that file. Hit OK. Then select the genotype file. You open, and there we go. Just as you saw before, we have the lines on the left and positions up top. Compared to the previous data set, which came from soybean, I believe, this one has a lot of colors, and that's because it's highlighting a lot of the heterozygous positions. The crop is intermediate wheatgrass, which is an outcrossing hexaploid species. And as an outcrosser, there's always a lot of heterozygosity floating in. This population in particular had about 40% heterozygosity. Sometimes it's as high as 60%. Sometimes it's a bit lower, depending on what loci you happen to sample and how the population was behaving prior to being genotyped. In any case, you can see there's one line, the 289, that's kind of bad. If you want to throw it out, you can. We're not going to deal with it right now. but I will show you how to do so. So for that, you want to go to filter, and then the entries or the lines are labeled as taxa for some reason. And then there's different parameters that you can use: heterozygous and minimum proportion of sites present. Let's say we want at least 40% of the genotypic data to be available. So this 289 hopefully will be kicked out once we do this. Others we're just going to keep at default values. Unchanged. Okay, that did not do anything. Maybe I need to increase it or something else. Probably there is genotype table taxa, something like that here. Let's make it 60%. There we go, it's gone. So probably needed a higher filtering value. Anyway, but we're going to go with the default, the base file, because that's what you have today with you. So now that we have these files, we're going to first take a look at phenotype and then move on to the genotype data. So let's click on the phenotype. Here we have the taxa or the entries on our left, and then I have 3 traits: seed area, plant height, and heading group. And this species kind of heads weirdly, where you have a plant, but even within the same plant, there's hundreds of tillers. About half, a little less than half of the tillers, they head very early. The others kind of gradually head later. So I take the data as in what group, or like in what phase these head. So 0 is probably, say, the first 7 days, and 1 is the next 7 days, and so forth. In any case, we have 3 traits to work with. So the first thing we are going to do is look at how the phenotype is distributed. Are there any outliers? You ideally want to do this before coming into TASSEL, but a way to do it here is go to results. Then you want to click on chart. So we are going to look at a histogram distribution of these traits. Let's say seed area. Looks fairly normal to me. And you can always change the bin numbers to make it more dense, or, oh, not 0. There, or 10, and so on. Looks okay. Then click another trait. Looks fine. The plant's going to be fairly tall, 175 centimeters, unless you work in corn or some other things that might be. Then you go to heading group. Overall looks okay, and that's because this is sort of data that I already worked with, so there's not too many outliers. But let's say that in heading group distribution, you see a value of 5, then you're like, okay, something's off. Want to go back to your phenotypic data file, look what's going on. If it indeed is accurate, leave it as it is, or delete it if you don't want that one particular line up there, and then re-import the data. Now we're going to go to the genotypic file and do some relationship analysis. The first thing we are going to do is, actually before that, let's do some summary of the data. So once the genotypic data is highlighted, click on data, geno summary, and we want all of it. Why not? Doesn't take that long. There you go. It's already done. Sort of gives you, the first table is a summary of what you have, how many taxa or entries are there, how many loci. I have a little more than 10,000 SNP markers here. The overall proportion missing, it's very low, and that's because, again, although the file name says 5%, this went through imputation already, so the proportion of missing is very low. And other things that you can see there. Then you can click on the allele summary. It'll take a while to load, unless you did not click on it. Okay, did not. Let's click on site summary here, and there's a lot of numbers: major allele frequency, minor allele frequency, major allele site, minor site, so on. Let's go to results and go to chart. We'll keep histogram, sure. Let's say you want to, this is wheatgrass, so there's a lot of heterozygosity. So let's say I want to look at how much heterozygosity is there and what's the distribution. So there should be a proportion heterozygous, and that's what it looks like. Ranges from 0 to 1. Again, highly heterozygous in wheat, which is inbred. this will probably be very close to 0. So you'll probably see a big bump up here and then almost nothing on this side. And depending on your species, this chart obviously will vary. Proportion missing, not much, already imputed. It's very low numbers here. Let's see, minor allele frequency. There's other things you can look at, but just the overall quick and dirty way of looking at how your data is distributed, both pheno and geno. And I would say this now, and I'll probably repeat myself multiple times. TASSEL is pretty bad when it comes to generating good figures for publication. So you want to use R to make better data. Once you have this, you can always export it. You could obviously also recreate all this information in R, but let's say you do it in TASSEL, in export, you can go to results, table, and you can do a CSV or a regular text file, then it'll save what you see on the screen as whatever name you give you. Let's say it's a site summary. Hit enter, and then it's saved. I don't know if I have the folder open. Let me see. Just 1 second, please. So here is the site summary that we just did, and then there's the Notepad++. It's an excellent program if you do not use it already. If you're on Windows, possibly Mac too, Linux people have it by default, I think, I'm not sure. There's another program called Vim, the Vim editor. And if you've used Linux, let me find the link to it and I put it here. You already probably know some of the functions. It's extremely powerful. Notepad++ cannot open files that are more than a few gigabytes, not base, in size. But Vim, you have 20 gig file, no problem. It'll take a few seconds to load up. Let me find the zoom here. There we go. Let's see, I'm gonna. Oh, maybe I cannot. I don't know. I'm very bad at zooming, guys. I'll add the link later to the GitHub page, but essentially this is what it is. VIM. You just go to Google Vim, and you can download. Looks like, yeah, for Mac too. Again, extremely powerful. Unfortunately, it will not have those buttons like you have for Notepad++. As you can see here, there's file, edit, search, view. It's more or less based on commands, but again, extremely powerful. Highly recommended. All right, back to this. So now that we have the genotypic summary, we're gonna go back to our genotype file. We are going to calculate linkage disequilibrium. I did not want to go through theory. That's why I might actually just skip. I have, as you can see, I'm going to pull it here. I have 4 walls of texts here. Most of you are fairly familiar, but I will have Susan upload this on the GitHub page. Just some background theory. I'll probably talk through these as I go along, but what LD is basically association between alleles at, genome-wide, but we mainly want to look at each chromosome. And it's not linkage. It doesn't have to be physically linked to alleles. So alleles that are even far apart could be in high LD, depending on the relationship. And let's see what that means. So you want to select the genotype file, analysis, and diversity, linkage disequilibrium. So the option is sliding window of 50 markers, or I think so, yes, or the full matrix. And if you look at the number, this is the whole genome file. That's a lot. It's not going to take that long, but I wanted to show you another way of doing it, because if you select whole genome, it will try to calculate relationships between loci in different chromosomes, and that's mostly meaningless in terms of LD anyway. So what you want to do is, with the genotype file highlighted, you want to come to data, and then you want to click on separate, and it'll automatically split into each chromosome. Again, it's a hexaploid with 21 chromosomes, so I have 21 files. The main file had 10,000 plus markers, and if you click on chromosome 1, there's about 500, 580, and so on. I'm going to delete all but the first chromosomes. So we have chromosome 1 here with only 500 markers. So with this highlighted, I'm going to go back to analysis, diversity, LD, and instead of now sliding window, because it's in a smaller file, I'll do full matrix and get all the relationships that I can get very fast. So in the output here, there's a few things. The ones that are of most interest are this right here, distance in base pairs. You can convert to megabase pairs later on. And then there's R square and there's D prime. These three are of most interest. But in the literature, you see R square, which is a correlation or the relationship between the two alleles that are, say, this base pair apart and that base pair apart. And that's the figure you see in the example. I think I have it pulled up here. And something like this sort of figure you have for different chromosomes. You have the sort of a decay shown here in the blue text. I do not think, unless I'm wrong, I do not think we can calculate LD decay in TASSEL. So I did this in R. If you are interested, shoot me an email afterwards and I can share this script. Basically, we're calculating the decay value, this blue text, sorry if it's difficult to see, based on like an arbitrary or widely accepted R-square value of 0.2. So for chromosome 8, it happened to be 5.3 megabase pair, where LD decayed to half of its value. But we will generate this figure in TASSEL, not just the decay value. So with your LD value highlighted, you want to go to results. Then you want to go to a chart. In the chart, we're going to switch to a scatter plot. So on the y-axis, we want the R-square value. So scroll down and click R-square. And on the x, we want the base pair. So if you scroll down, there's distance, base pair. Take a couple seconds. So there we go. Very similar looking file as you saw. It's a bad looking file for publication, but it does the job, gets the job done right now. And this is very similar to what probably you've seen in a lot of papers. Funny thing, you cannot close this. So you just got to minimize for some reason. TASSEL is interesting. So then you also, to export, you go to results, table, and export a CSV or a text file. There's another way to export as well, file and save as, but this is a bit more, sometimes if you're not in the same folder, I think it puts in the document something weird. So I'm not going to save this. One thing you will notice here is there's a lot of these NaN values and these square blocks that it could not calculate. So these you will want to filter out, and it's much easier to do in R or even in Vim. There's a one-line command to throw those out, so you have only the meaningful numbers in your output file. Any questions so far from anyone? Okay, moving on. We are now going to look at, I think, PCA. Now this time we are going to select the whole file, not chromosome 1. Just take it out so it's easy. And we want to do PCA analysis, which is just looking at, oops, sorry, there's something in the chat. Man, I should take like a TASSEL, Zoom tutorial. Hannah, can you please guide me how I open the chat, please?
I can just ask it here too, that's fine.
Yeah, sorry. thanks.
I was wondering why if there would be a reason to use like sliding windows of different sizes versus the full matrix LD calculation. Like, I don't understand the benefit of one versus the other besides like saving processing time or whatever.
Yeah, I think that primarily is it. And you also, that would be number one reason. A second, also important reason is that if you have a big genotypic data set that has not been filtered for SNPs that are located very close to each other, and you implement a sliding window, then it's going to look at those different windows, and you'll have a more, say, a meaningful relationship calculated. Whereas this particular file, I think, was already filtered to remove SNPs that were, I can't remember, 1 kbp or 1 mbp of each other. But my understanding also has been to mainly save time. But even if you were to do a full matrix, it does not take that long, but the output file that you save is going to be quite large, that will definitely need R or Linux to open, or Vim. I think mainly for the ease of operation later on, and to not retain anything that you would not need right away. So unless anyone has any thoughts on that.
Thanks.
I have another question. Mm-hmm. So why was it not able to calculate the LD for those ones that came up as squares?
That's a great question. I do not know, I'll be honest. It could be because there is no relationship. Sometimes that happens. Like if the loci are very far apart, maybe there is no meaningful relationship to report, and that's been my understanding. Or that they're so close to each other that they're essentially zeros, and I do not know if it does zero or makes it, say skips that value. I'd probably have to look into that and get back to you. Actually, I've just taken it for the face value that, okay, if it doesn't, then I trust what TASSEL does, so I'm not gonna worry about it. That's been my thinking. That's a good point.
Okay, thanks.
Anyone else? Okay. All right, on to PCA. So with the whole genotypic data set highlighted, we want to go to analysis, and this time it's relatedness. We have distance matrix, kinship there, MDS, PCA. I've never done MDS, but I've always thought it to be very similar to PCA. But for today, we'll do PCA, and we'll come back to kinship at a later point. In scientific papers, you see, or peer-reviewed papers, there's 2, sometimes 3 PCA axes reported. TASSEL by default generates 5. We'll keep it as it is. Hit OK. It'll take a few seconds, but it's not too bad. All right. So the first table it shows is the PC values. That this is the one that you report. So usually PC1 is pitted against PC2, and sometimes PC2 against 3. And then the second table is the eigenvalues and the proportions. So when you look at this kind of figures, you see what proportion the first axis and second axis explain of the whole genotypic distribution. And the answer is right here on here. Proportion of total is the first axis. It says 0, but that's the first one. That's 3.3%. The second one is 2.6%. And if you sort the cumulative, it's 1 all combined. And if you look at the number of PCs, that's it does for each entry of each taxa basically. So there's 450, but if you remember, we had on our genotypic file we had 451. So it starts at 0 and then goes to 450. So again, that's the number. Oh yeah, okay. So sorting is not 1 to 10. Interesting. All right. It goes through 1, 10, 100. But that's the number you want to report next to your X and Y axis when you report PC1 and PC2. The figure that I'm talking about is something like this. You go to results, chart, and we want to do a scatter plot again. And on the x-axis, we want to have PC1, and the y-axis, we want to have PC2, and that's the figure. Again, do not use this figure, use R, but that's what the distribution looks like. And if you want to do 2 versus 3, say we want PC3 on Y and PC2 on X. And if you look at these, the proportion of total again, sorry, it kind of like disappear here, number 2, I find it. So the first axis is only 3.3 and the second one is 2.6. So combined, the first 2 axes explain less than 10%. And that was typical of our population, but in your populations, you might see the first 2 axes might explain 30%, you know, like a big population structure occurring there. In ours, we found not much evidence for structuring. So the K equals 2 here, the Q matrix that I talked about, there was a very weak evidence. But in some cases, it might be strong, some weak, depending on what you have. Again, to export results, table, and then you export, and then you can import an R for better figures. I use ggplot2 myself. So any questions on that? All right, moving on to kinship now. Very similar, you click the genotype file, you go to analysis, relatedness, and then kinship. There's different methods here. You can click on help and it'll provide you explanation here and the papers, what they deal with. And this is centered, I think, identity by state. And I'm going to keep at the default values, hit OK. It's very quick. And you see a square matrix of the upper and lower triangles, each line and the value with other lines. And I would say here that these numbers are relative. So for example, you know, you might be wondering why it's not 0 to 1. I do not know the answer to that, but you can convert to 0 to 1 if you prefer to, as it does make filtering a bit easier later on in some of the other analysis you might do. But these numbers might be different in your population. What you want to do is, if you want to look at the, say, relationship between the whole lines in the whole panel, we want to export it, import an R, obviously, and do a heat map and see how the lines are related to each other. And again, for that, click on results, table, and then you export as CSV or tab. All right, we are going to now switch to GWAS now that we have calculated LD, PC, and kinship. We're not going to use LD for GWAS, but that was just a different analysis I wanted to show. Any questions we want before we jump to GWAS? And yeah, please just speak up if you have any questions. It's easier for me to, because I cannot find the chat button. So TASSEL gives you 2 options of for doing GWAS, genome-wide association scan or association mapping. If you go to workflow, you can see there's this GLM that has either PCA or Q. Again, PCA is the PCA analysis we just did. Q is the Q matrix from structure. MLM, so both Q or PCA plus K, which is K is for kinship. GLM here stands for general linear model. It can take the genotypic effect and 1 extra fixed effect, the Q or PCA. MLM is mixed linear model. It can take, of course, the geno plus the Q or PCA, and then an additional random effect, which in TASSEL's case is kinship. And if you click on workflow, I'll not do the workflow, I'll not follow this one, but I just want to show if you have all the input files ready, you can click on, let's say, GLM PCA. And then the other window opened in another screen, but basically you click the genotype file, the phenotype file, the PCA file, so one at a time. If I hit cancel, it'll ask me to open it next time, another file. I'm not going to do it. Maybe I can do it right now, right here. So first you load genotype file, and once that's done, it'll ask you to do a few steps, and then you open your phenotype file and so on. Like basically a step-by-step method to do the association mapping. But here I will skip that and show you how I typically do it. So in the analysis tab under association, we have GLM and MLM. There's also weighted MLM. We're going to skip that for now. And earlier we talked about genomic selection if you wanted to do that, which is an entirely different thing. So if you click on GLM, it'll say something's a data set that can be analyzed by GLM has not been selected. So for this, TASSEL needs certain files in combination. And for GLM, those are your genotype. And then with the control button held, or command in Mac, I think, you want to click on the phenotype. And then scroll down. Where's our PCA? No, there it is. PCA file, I think that's it. Then you go to data, and then you do intersect join. It'll create like a combo file. You can see on the screen, we have our entries or the taxa on the left. We have the 3 traits. We have the 5 PC values, and then the genotype also there. That's the minimum requirement for both GLM and MLM. But for MLM, we're going to bring in the kinship layer. So here, once you have this done, again, geno plus pheno plus PC, combine, data, intersect join, and you'll have this file. Then you want to go to analysis, association, GLM, and then we get this window here. I'll keep everything at default, but there's a bunch of things you can do. You want to save whatever it generates automatically, you can do that. Usually you want, about 1,000 permutations or so. I'm going to skip that for now, just so that we're done with it quicker. If you do not want to use anything but biovidic sites, there's another thing. But again, everything at default. There's also a help tab right next to it if you want to go through the filters and the parameters. Hit OK. GLM is pretty quick, just a few seconds, and we are done. So it generates us 2 tables. One is the stats, the overall your marker and p-values and additive and dominance effect and so on. The second one has your olytic effect estimate. This one we're not going to do much today, but let's come back to the summary on the main big table. First thing I always look at is the QQ plot, which basically shows how are the results. Is there any bias that was introduced because of population structure? So for that, you want to click on results, QQ plot, and we're going to plot all 3 traits. Why not? So this is what a QQ plot looks like. Your expected is that straight line if everything is, say, working okay. The green one is very close to the expected line, which is the heading group. So probably not so much of a problem there. The blue and the red, the other 2 traits, there's I can see there's a lot of type 1 error, meaning basically a lot of false SNPs that were called as or called to be associated with these 2 traits, which is not true. And MLM can fix that issue, but at least I wanted to show you the difference between these 2 methods. The tail here, this portion indicates significant SNPs, and while it does so, again, the farther these lines are from the straight line, the more error there is in your analysis. So we are going to, I'm just going to close this for now, but we'll come back to it once we have the MLM done so we can do a side-by-side comparison. with the same table highlighted, let's look at the Manhattan plot, which shows you genome-wide SNPs that are, here, that were significantly associated with the trait. So you cannot look at all 3 traits here, unfortunately. You see the area, okay? Excuse me. So it looks like we have few hits. But again, do remember that these were done without any permutation and not a strong control for population structure. So, but nonetheless, you can see that on a few chromosomes, there's a few hits. Looks like in chromosome 11 here, there's a pretty big one for seed area. And same with chromosome 15 here. Now, will this hold when we do MLM? We'd have to find out. Then you can save this plot just for your, just a quick and dirty way of looking at it. There's different options, but it's not worth it because TASSEL does not make good figures. Then you go to results again, Manhattan. Let's look at height. Okay, another traits, a few other hits, and so forth. And you can again import all of these in R for making better figures. It's in my notes here if I'm missing something. Well, let's do a side-by-side comparison with MLM then. Any questions in GLM? Maybe we can actually hold until MLM is done because the questions might be quite similar. So then you have this combined data set and you want to do MLM. You go to association, MLM, it's gonna say no, because as I said, MLM requires kinship matrix. So then with this highlighted, you click on the kinship matrix we estimated earlier up there. So once both are highlighted, this time you do not have to do intersect join, just that is enough. Go to analysis, association, and MLM. Now a few options here. We're not going to change with anything, but the main, I guess, option I want to discuss here is this variance component estimation at once or for each marker. Some people do each marker because that apparently adds some power to the association mapping and helps control some false positives. In my opinion and experience, the largest QTL will be present no matter what. It will change your minor QTL. Some might reappear, some might disappear. So you might have to do it, once without and once with, just to see the comparison. But we're going to keep everything at default for now. We're going to run. You can write to an output. You can filter. So you don't keep p-values larger than a certain threshold. I'm not going to do that for now, but there's an option in case if you want a smaller output file. Hit OK. This will take a bit longer than GLM. Maybe any questions in the meantime, because this will probably take about a minute. While that's running, on my notes I have, so both GLM and MLM are single locus models. What that means is they're going to analyze one marker at a time, report back to you saying if it's significant or not. But if you think about certain traits like, say, yield or even height, perhaps, there might be complex traits where there's multiple genes, multiple loci controlling the trait. So analyzing one marker at a time is not the best approach to do so, is what some people say. So there are other options where you have multi-loci models. And one of the common ones you might have heard is the farm CPU. There's also, I think, MMLM, the first M for multi-MLM, something like that. But there's a few options that you can explore. And they both, the MMLM and farm CPU are both, I think, available as R packages. If you wish to say, at least for a couple of traits, you wanted to try just MLM versus a multi-loci model. Okay, almost done.
Does anyone need Praveen to retrace his steps on any of
Yeah,
the things we've
sorry.
done thus far?
If I'm going too fast, please stop me. I can go through this again too if needed.
Oh, we have one yes.
Okay. Yeah, let's compare the results and we'll go through all of these again.
Okay, thanks.
Clean slate. Yeah. Okay, it's done. We have some residuals. Ignore for now, because mainly we want to look at, and here's allelic effects, obviously. But this is the big table from MLM. And the first thing, again, let's look at, go to results, the QQ plot. We're going to keep all, why not? So I don't know if you remember, but this already looks a bit different than the previous one. So I'm going to go back to GLM, make the QQ plot. So on the left, I have GLM, and on the right, I have MLM. So for the most part, the lines are now very close to the expected, which is a great sign. And this, as you know, because we included kinship to control for population structure. Oh, before I forget, if your line, you can see the green line now in the MLM method in the plot on the right here now is under the expected one, whereas on the left one it's over. What does that mean? Well, if it's over the expected line, it means that there's chances of more false positives being introduced. And if it's under, the chance of false negatives being introduced. So it's like, okay, then which one is the best? That depends on your data. That depends on the analysis. You might want to stick for height. Sorry, the heading, green is heading, with GLM, whereas for the other two, it may be MLM. But that said, usually when you write a thesis or write a paper, reviewers prefer that you stick with one method because then you're not introducing this method bias in your analysis. So if I were you, given looking at the data, I'd probably stick with MLM here, because I can say that, hey, we control for population structure. Sure, there's looks like some false negatives being introduced, but it's better to have fake QTLs than lose some. But again, like I said, that depends on what you're doing, your trait, and so on. Another takeaway from comparison here is that on GLM, despite controlling for population structure, height was okay. And on the MLM, when we control for structure, it kind of like went under the line. Again, not necessarily a bad thing, but that also probably tells you that maybe you're overfitting your models. So that is another sign to not use MLM for this trait. However, if you're using like 7 traits and there's like 2 under, others above, or the other way around, then I guess it depends on your discretion on what you decide to go forward with. But at least for the most part, I'm more happy with MLM than GLM. So with that said, now let's compare the Manhattan plots. We're gonna do GLM with GLM highlighted. Go to results, Manhattan. We'll do seed area. Keep this to the left. MLM, highlight that, results, and Manhattan. We're going to do seed area. Okay. Keep these to the right. In the GLM method on the left, we had 2 big ones. Chromosome 11, this green one right here, and then this dark gray chromosome 15 were quite high up there. But on the MLM method in the right, they are pretty much gone, except, well, on chromosome 11, one of these might be the same marker like these 2 right here. And on chromosome 15, there's a couple here. Like I think some of these might be the same ones here. What MLM does is, by taking the kinship into account, relationship among the lines, it does control for inflation of the p-values, and that's why MLM is a bit more preferred than, say, GLM. But you have to export the data sets, the outputs, and then see if indeed these markers are the same. They probably are, again, talking from experience. And if you look at chromosome 2 here, we had, these markers around LOD of like 5, around 5. And again, I think these are the same markers, a little bit controlled for the p-values. So if you remember earlier, we did not do any permutations. And a reason why you want to do this is, let's say you pick the widely used LOD value of 3.0. On the GLM, if you draw the line here, you'll have probably 50-some QTL for one single trait, that is. Some of these might be co-localized, that's okay. But a lot of loci that are supposedly associated with this trait, which might or might not be true. Even in MLM, it's not highly controlled. If you draw the line at 3, you still have about a dozen or so markers. I'm not saying it's not possible to have that many loci being associated with the trait. It might as well be, but just to make sure that you aren't reporting anything wrong or false associations, if you introduce, if you use the p-value, if you use permutation, then the program will suggest you a p-value that you can use as a threshold to report anything above it. So we're talking hypothetically here for GLM. Had you done that, maybe the program would have suggested that your p-value after permutations is 5.5. Then you'd have, well, only 2 loci that were significant. And that's another way to kind of control how many loci you're reporting that could or could not be false associations. Let's look at another trait before we move on. We're going to do MLM again, go to results, Manhattan. Let's look at heading, okay. Keep to the right. Go to Manhattan, heading, okay. I'm interested in heading because if you remember, the QQ plot showed for GLM, it was pretty good looking for GLM and not, well, a little under the line, but not too bad for MLM. But let's see. Well, the first thing to look at is the LOD value. So this one, it goes as high as maybe 4.3. Here, it's a little controlled, no more than 3.4. But as for the number of, if we were to say shrink this down a little bit, the markers that are appearing up top here, they look about, in the same positions as here. Of course, that's not an indication without knowing the SNP names or the loci names. But again, speaking from experience, I do think that these loci are the same. So this, despite the heading being a little under the expected line in the QQ plot, I think we have the same loci here. And an easier way to test this, of course, is go here. So we have this big table. We are going to look at, yeah, it's a little tricky. Let's look at p-value. P-value of, negative, the smaller the p-value, higher the LOD, as you know, because it's a logarithm of p-value. Trying to find heading group, just one to find as an example. So it looks like we have one, sorry. On heading group, we're going to do a Manhattan, just so make a mental markup. Okay, chromosome 9, this maroon colored one, was pretty high, around 4.3 for GLM. And looks like for heading, we have chromosome 9. That probably is the same marker that we saw in that plot right now. So that's, around the 9883704. Let's go here. Again, this is very archaic way of doing this, but I just wanted to check. So we have a heading group here. That's chromosome 19, but if you come to 9 and look for heading 9, that's the same marker, if you remember, 988037004. So those markers appear with different p-values, obviously, because of the methods we implemented, but they both appear to be significantly associated with this trait. So again, despite the line going a little under the QQ plot, I think we're okay in terms of the marker being significant for this trait. Not too much to worry unless it deviates a whole lot from the expected value. Okay. Next, I want to talk about the p-values here. So in TASSEL, I do not think you can correct the p-values for a false discovery rate or a Bonferroni method that, as you might have heard. What you can do is export the output table, and in R, if you just search for R package Bonferroni or FDR correction, there's few that will pop up, and you can import the data and based on the size of your data set, you can correct for any inflated p-value, and then report the ones that come out as significant. It's if you use if you do GWAS in R already, for example, I like to use GAPIT because it's essentially TASSEL but in command line format for GWAS. I think it already outputs you FDR corrected p-values, and if not, I'm sure PharmCP probably does it too. I've not used it. And there's also other packages that can do that for you. So you're reporting only the correct significant QTL. All right, questions then, because I think that is all that I wanted to show you and might have run through very quickly. If there's any questions, let's go through them. And after that, maybe I can run through the analysis one more time, depending on where or what steps you might want to see.
I have a question about the imputation
Mhm.
that you did. So intermediate wheatgrass is an outcrossing crop, right?
Correct.
How well does the imputation work for you?
Pretty good. I will have to pull up the exact number, but it's above 90%, close to 95. Sorry.
Yeah, and also I guess how do you look at your data and like how do you determine how confident you are in the imputation, especially if you have like very heterozygous and like outcrossing crop?
Yeah, so this was the data I'm showing you here was something that we'd already published. I'm gonna just show you because I don't remember as much. I typically filter for 20% or less. In that case was, well, this is again, like I said, for the workshop, I refiltered to have only 5% missing data, but on a study we published, it used 20% missing data, and then imputation doing the LDK and NI, you saw it before. And yeah, we had imputation accuracy of about 95%. That gives me a pretty good confidence in whatever I see as being a true positive. Of course, until you validate, in a separate individual population, you cannot say that, but because of the rigor that was implemented, at least I was, and I still am fairly confident in the work that I do. You do have to be careful. I think you had a question earlier about having a non-reference based SNP calling. Is that true? Just to confirm.
Yeah, that's what I'm going to be working with.
Yeah. So especially well, I have not used many programs that use non-reference based, but I have some familiarity with TASSEL or the unique pipeline, as they call it. And it does tend to introduce a lot of errors. So in that case, you might want to say start with a data set that has the least amount of missing alleles as possible without losing your coverage of the genome. And after that, anything above 90% imputation accuracy, I think, is pretty good. I had a paper published many years ago in wheat where alleles, and that was by print, not a GWAS panel. But in that one, missing data of up to 30% in dire situations was okay, but anything above that, it started inflating the linkage maps, the LOD values. So it definitely introduces a lot of errors. So as low as you can keep it, the better it is.
Okay, thanks.
Any other questions?
Sorry, if no one else has one, I have another question.
That's fine, yeah.
So I saw that you calculated the kinship. I also saw that there's an option for calculating a distance matrix. So what is calculated exactly in that distance matrix?
I think you have to look at the definitions of those. Kinship is, there can be sometimes negative values. Again, these are relative numbers. They can always change. Distance, probably not. We're using the same marker set to look at relationship between the lines in the same set. So if you were to do correlations between these two, I bet that they are, you know, highly correlative, but I have not done that, so it would be very wrong of me to say, oh yeah, they're the same. Maybe the error will be, you know, when you try to use distance matrix as a covariate in your MLM model in TASSEL, it will say no. I have reported distance matrix in the past, mainly because it was very easy to do so using the VCF file. There's a program called VCF Tools. I did it fairly easily on the MSI, so I don't have to play with it later. But if I had to take a guess, I think they're very similar. Maybe others in the group have some experience. I don't do much genomic this distance, only kinship and PCA.
Yeah, thanks.
All right, well, I think someone wanted to go through some steps. Can you please tell me which ones, or all, or what do we do? Well, no response, but we have time, so let's go through all. So I'm gonna select all, get rid of all. Yes. So let's import our files. Okay, so this time we're not going to do the hap map. Best guess we'll do as hap map later. For now, we will take Pheno. We're not going to do Q matrix, so we're not using it. Open. So that's our phenotypic file. Now we're going to open our genotypic file. We go open as, and we already know it is a hap map. If you have VCF, of course, there's a VCF. And then your sort position is always on, although it's not needed in mine because it's already sorted. You click on OK, then you click this, and then open the genotypic file. Here it is, imported.
Hi, Praveen. Just one little thing to add. I'm suspecting the reason the HapMap file doesn't open automatically.
Oh yeah, that's right.
Is probably because in the file name before HMP there's underline. But if you change
okay.
dot, that might work.
Let's give it a try, guys.
So if you change, the file name, yeah, to dot.
So okay, now we're at, well, let's make sure we're at make best guess, so not HapMap, and open this. It might say it's already open, but let's check. Oh, never mind. Open. Yep, you're right. Thank you.
Yeah.
Wow, that's a great
Yep.
tip. Okay.
Welcome.
Yeah, let's take this one. So to filter some of these, like that 289 that has a lot of missing values, go to filter. You can also filter the alleles themselves. There's a lot of options. You can buy minimum count, allele frequency, what max heterozygous you want. Right now it's 100% because again, I have a highly outcrossing crop. But say you wanted to limit this to, I don't know, 20% heterozygosity. So you can see there's a lot of A's and T's and C's and G's and less heterozygous loci, and from 10,000 some SNPs, we drop down to 2,500. You could do that, you know, if you had a not outcrossing crop. So I'm going to delete this. Then you go to filter. Now we're going to filter the entries. And we said, you know, without taking count, I don't know how much 289 has. So we'll just keep as each entry has to have at least 60% of all the loci present. And maybe this has 50%. That's why it's being thrown out. And you can also do heterozygous here and so on, but I'm keeping those minimum and maximum as it is because outcrossing. Oh, if you want to manually delete some of the lines, you can have a file that has, let's say you want to throw out 10 lines. You can have a file with each entry in a single line, I believe, and then you open that and then hit OK, and it'll kick those out too. For now, we're just going to do the minimum proportion of size present. Okay, that's gone. If you go, that's from 451, we drop down to 450. It threw out just that bad guy. So let's delete that one. So we'll work with the filter one this time. First thing to do is LD, diversity, linkage disequilibrium. You click, and let's do the whole genome this time. Let's just see how long it takes. And you can, you know, do more or less, and keep it as default. Now here's a kicker for me: how to treat heterozygous. I did not change anything in the previous one, and now that you've seen I filtered the heterozygous sites, you might be wondering, aren't you losing a lot of lines? Loci and information if your heterozygous calls are set to missing. Well, unfortunately, TASSEL, that's how it operates. I could treat it as third state, but that's technically not true because my species, just like wheat, despite being a hexaploid, behaves as a diploid. So it's not a true third state. And I can ignore or do missing. And this, I just keep it as default because I've always done that. And in the analysis or the results you've seen, the plots look fairly similar to what other wheatgrass geneticists have been reporting. So I've been just going with, okay, it works, why mess with it? But something to think about nonetheless if you also have an outcrossing species. Not full matrix, it'll take a while, so I'm just going to do this fairly quick again, even on the whole genome. And as you saw, we have the same table. Go to results. Oh yeah, well, another thing did not show previously. You can do LD plot, but it's a very ugly looking plot. You can sort of zoom out to see, you know, the all-off relationship, but it's just bad. Not good at all. Close it. Then we go to chart, scatter plot. On the y-axis, we want to have R-square values. On the x-axis, we want to have the distance values. And oh wait, this is the whole genome, so it might take or not. This is the whole genome. If you already have the chromosome information, you do not want to do this. You might want to, but it makes less sense to me when you have when you can report values in each chromosome. You can have like more detailed conversation with the numbers shown for each chromosome. But if you want to look at the whole genome, this is one way. Well, you can save it, but why bother? I don't know. I think the buttons are not working on this plot, so yeah, let's minimize. My TASSEL might be frozen. Yeah, that was another. I think someone asked why. This is why. Another reason to not do the whole thing. Might have to bring in the Task Explorer here. I'm sorry. There it is. Okay, back to TASSEL. just open this time. I'm gonna do this. Perfect. And then right. So next was, well, let's throw it out. PCA, relatedness. Just want to show you MDS because again, we have some time. Number of axis 5, sure. Distance matrix as input. Oh, interesting. You can see how we've never done this before. Okay, there's your distance matrix. And then I think you do control, select, relatedness, MDS. Okay, right, there's the MDS. Again, like I said, it's very similar to PC because it actually reports as PC, so sure. But if you generate a figure, it does look different, and I'll show you. So next, we're gonna do our actual PC analysis. Go to PCA, default 5-axis. Okay, it'll just take a few more seconds. All right, so we have the values, some values here. I don't know if we can compare side by side. Probably not. Let's just look at the scatter plots because you will see that they will look different. So for the MDS, we're going to do PC1 versus 2. This time we have PC2 on x-axis. Select the same for the PCA. Scatter 1 on Y and X. Oh, I don't know what happened to the other plot. Wow, this is interesting. Oh, there it is. If you look at the axis, they're different. And not just that, if you look at the distribution of the dots, they're different too. But I think it's just because the methods are slightly different. I have not used MDS in my GWAS with TASSEL, so I cannot tell you if the results are going to be different. But again, if I have to take a guess, probably not a whole lot difference there because MDS reports these as PCs too. And if you look at eigenvalues, oh wow, those are highly different. So they do add up to be more than one. So interesting. Yeah, my recommendation would be to stick with PC values if it works. Why switch to something that you know that is kind of weird. All right, now that we've done PCs, we're gonna do kinship. So go to analysis, relatedness, and kinship. Everything as default. Hit OK, and it is done. So for the GWAS steps, we have genotype, we have phenotype right here, and we have kinship right here. And then we have the PC values, so we're ready. Now for GLM, we want to select genotype with the control button held because you want to select multiple files. Genotype, hold the command button, click on the phenotype, and the PC values. I don't know if the order matters, but that's how I've always done it. You go to data, intersect join, and we have a file that's combined all those values that we wanted. So we have the traits, PCs, and the genotype. Once that's done, you want to go to analysis, association, GLM, permutations. I don't know how long it'll take. Let's try 100. Hit OK. So as you can see, it's progressing slower, and that's because, of course, the permutation of the trait values takes a while. If you have more traits, it's going to take even longer. But I think it's going fairly fast that we can wait and let it complete. it's done, of course, the first thing to do is, oops, results and QQ, everything. Looks like, you know, of course, it's the same. It doesn't do much different than the previous one. We want to put in Manhattan. We want to do C area. I'd say the results look fairly similar to what we obtained previously. So maybe the permutation of 100 was not enough, or I'm trying to find. And there's a permutation tab here. There's, you can see the p-value permutations. Typically, the lower numbers tend to go with the lower p-values. You can see, you know, if it's high, then it's also high. But there's nothing of significance here, unless I'm interpreting it wrong. But I've typically done permutations of 1,000 even higher, and that would be another reason why you want to consider running this in command line. It's typically faster. You can run on MSI, I believe, as long as you have all the supporting files and have the proper Java version loaded, and then just let it run overnight or something. Here's two columns that we did not go over previously. The dominance. For my species, because it's outcrossing, this is a somewhat, you know, interest to me. From our experience of these traits, we know that dominance is not a big player, but nonetheless, it helps to have these values outputted. For wheat, my guess would be these are very low and with minimal effect, but in some cases, you know, these might be slightly higher than the additive values. Just something to keep an eye out for if you have species like mine where it outcrosses. Oh yeah, then to export, then results table and export as a tab or CSV, or you can also go and save as whatever works. Now to MLM, we select the file that had, you know, everything combined. And then with the command button held, you want to click on the kinship up here. Oh, you can actually, by the way, once it's highlighted, click again, and you know, if it makes it easier to track these, just name it to kinship. It's easier to, like, you know, go through these, then maybe scroll down combo for GLM. Then you know that if you have the combo for GLM plus the kinship, then you're good for MLM. So then you want to go to analysis. Again, you do not need to join these two. Association, MLM. We could try that, but I'm afraid I have to do you know, killing the program and reopening it. So I'm just going to leave it as default here again. Now, if you do gap it, by the way, you can also do all these things, and it's much faster than TASSEL, I think, with the added advantage. Again, you can run in command line, run on MSI, or even your own computer overnight. You don't have to monitor it. But for now, we're going to keep it default. Run. Let's filter this time. So we don't want anything larger than negative value of -3. Again, smaller the p-value, more, you know, stronger the association is. And this will take some time. This would probably be the last thing I show. So, and we will have a Q&A at the end, but any questions so far, or any concerns, anything that we can help with? While that's running, let's see if I can do something here. So let's open something in the Vim, as I talked about earlier. Right click, I have it right here, open or edit with Vim. It opens like this. Again, you do have these things, but command line is so much faster. You go, you wrap it, and this is just a demo. You don't have to follow or anything. And if you'd like to use it, just let me know, email me later, and I can help you out. Then that sort of puts it in like a tab-based format, easier to look at for your eyes. You can do page up, page down, why not? And then you can select with a mouse on Windows. You cannot do that on Linux. Well, you can, it's different. In any case, if you hit a colon, you can do a few things. Let's say that I want to change everything as NA. This is just for demo. The missing values here to BB, because that would be something I don't have here, and that will be very obvious. It's very quick and easy. You search for NA, and you want to make those into BB. Global means G for global, all file. And it's extremely fast. And this is not a big file. This is only 14 megabytes. But if you have files that's 5, 10 gigabytes, it'll do this in a matter of a few seconds to a minute, depending on how powerful the machine is. I highly recommend it because it saves a lot of time. You don't have to tinker with a whole lot of things. But we don't want to save it, so we're just going to exit. But I just wanted to show you that this program is my preferred way of opening these text files and working with anything. Search, replace, move around, those things are very easy. Right. Here the MLM is done. Again, we want to look at the statistics. And if you look at this table, it's much smaller because it filters out the non-significant ones. Well, why is it bad? It's bad because if you want to draw Manhattan, let's see what happens. You do not get the full marker list here. Like, everything is blank down here because, well, we threw this out. So for that reason, you might not want to do this, but for a quick copy paste or quick saving of just the significant, and you don't want to sort later, it's a good idea to do. Keep the filter on and then only get the significant hits. But again, just looking at only the significant ones, there's a few, I think they're probably the same as the previous one. What happens if you do QQ plot? We'll do all. Again, it's changed because we don't have markers. We're basically losing. This is the tail that you're seeing, the ones that are significant. Anything that was sort of following this line on this part previously is now gone. So filtering would keep only the significant, but we'll throw out more information if you wanted to look at that. If you look at height here, let's look at these two loci. They are both 162 megabases apart, only, well, at 162 megabase, yes. But if you look few kilobases apart, now, are they the same QTL? Are they different QTL? Well, depending on if you know how much you know about your species, you can declare that they are the same, but a more easier way would be to use other programs to declare if they are in the same haplotype block. One common program you've probably heard of is HaploView. You can import your genotypic data, and it'll, depending on some parameters you enter, it'll calculate the haplotype blocks in that chromosome, and it'll give you a list of where these SNPs fall. And based on that, I typically declare, okay, these two are most likely to be the same QTL because they're in the same haplotype block. So, this list of, I don't know how many there are, let's say 40, it'll probably trim down to about 25, 20 total QTL based on their position. Further down the list right here, there's on chromosome 17, we have the same marker 3 times for 3 traits. Looks like, oh, never mind, it's NA. It does that sometimes. I don't know why it shows here, but there's nothing. Sometimes, but the point was, sometimes you have loci that are common between the traits, and it's typically a good sign if you have closely related traits like yield and seed size, or even sometimes heading and yield, because does it mean that if it heads early, this is going to give you more yield, so on. But typically you want some loci that are shared between the traits or between the environments. If you have multiple environments, the traits I have here are from a single environment. But just different way of looking at things. And I have only 3, so based on the number of environments, if you decide to average your data or not, or calculate your blues, you might have more traits. Again, if you need any help, because I've done GWAS a few times, and I'm sure others in the group have done it too, or any help with the genotypic data conversion, any other things, Shushan is here, I'm here, feel free to reach out. But with that, I think I'm going to stop. I hope that was a bit slower than before. If there's any questions, we can take them now, and we can continue to communicate through emails as well.
I was wondering if you could mention that there was some R packages or something for
Yes.
multi-locus models.
Yeah, let me, so this one, this slide should be up on the GitHub page, but here there's PharmCP is a well-known example. Again, please don't quote me on this, but another is MMLM, but I'm not sure if it's our package, but I do know that the program exists. It probably is. Looks like there's something called MLM. I'm not sure if it's the same. Yeah, multi-locus GWAS study. So there's options, but PharmCP is by far, a very popular and very well-cited program. So I'd be very comfortable recommending that.
I have a question.
Mm-hmm.
Have you used TASSEL for this diversity analysis function?
Oh, not TASSEL. I have used, I think I've used the VCF tools because I calculated pi using that program. The numbers have to be the same, I think, but I'm not sure. I'm not in TASSEL though.
Okay, yeah, and I guess that is an example to follow up. Are there, for any of the things that TASSEL offers or that we've talked about today, are there, other programs that are better than TASSEL in these cases, besides data visualization?
For GWAS, I look at TASSEL first because I do all my imputations there. It's very quick compared to other programs. But then after that, I move on to GAPID because it produces, once you have, the script set, it produces files that you don't even need, meaning that it gives you all the information. You don't have to redo the analysis at all. It's all there. And it's essentially TASSEL because the postdoc who, well, is now a faculty at, I think, Illinois, he used to be at Cornell, not sure if it was Buckler Lab, but they do very similar analysis. I want to use PharmCP, but have not gotten there because I haven't done GWAS lately. But that's another very common. People also use RBLUP for quick and dirty GWAS. That's another commonly used program for both GWAS and genomic selection. Sushin, do you have any recommendation for the part of your presentations?
Sorry, it's not my specialty either.
Okay.
Yeah, anyone else who have experience might want to share?
Yeah, to reiterate what I said earlier, as long as you have big QTL, major effect QTL, I think essentially all programs will find it with ease. It's the minor effect QTL that some programs might do a bit better job. Maybe the multi-locus models would be better at those than just MLM, or maybe they will overfit the model. I don't know. But I'd have to say that, yeah, like I've seen even using GAPIT and TASSEL, the small effect QTL, they reappear or disappear. And it's, I don't know, I at the end of the day, I just stick with one program and report everything it shows. Some people also compare different models or different programs even, and only show the ones that are common across, a few. But given what Rex Bernardo said in a paper a long time ago, most QTL reside in the literature anyway, so I don't know what it exactly means, especially for the minor effect ones. Another reason why genetic selection, is more popular, because at least they can be included because of the marker effects.